All right, so this is the first of probably two podcasts on cell energetics or cellular energetics, and this first one is about cellular respiration. Um, and so here are a couple of the targets that we're going to be able to do. I added a couple things. I updated a few things, and obviously I didn't write it all out. So first, we're going to talk about the definitions of cellular respiration and make sure that we understand ATP and what ATP is, and why it's so important. We're going to talk about glycolysis, anaerobic respiration, aerobic respiration, but we're going to do this in the order of 1, 4, 3, 2. So we're going to work backwards a little bit here. But let's talk about cellular respiration and make sure we understand what ATP is. Oops. Before before we move too much further along through the process, so you understand what the goal is. So let's talk about the definition of cellular respiration. So cellular respiration is the controlled release of energy from organic molecules, and that energy comes in the form of ATP. And so that's what really you want to know. Cellular respiration is different than, let's say, combustion or burning something because it's not controlled. In cellular respiration, there's a very systematic way in which chemical bonds, covalent bonds, get broken, releasing energy and creating ultimately lots and lots of this molecule, ATP. And when we say controlled, maybe I'll just point out what I really mean by that is that it's enzyme. There are enzymes that control this reaction, as just any other reaction in an organic or in an organic pathway. So enzymes control that reaction. So that being said, let's talk about ATP. By the way, this definition is maybe something you need to memorize. When I say maybe, definitely. So let's talk about ATP. So ATP stands for adenosine triphosphate. So we've talked about some of the molecules that have phosphate in them already. You should recognize that phosphate is part of the phospholipid molecule that fits into our lipid category. We'll talk more about that when we talk about cell membranes. We also see phosphate groups in our DNA structure. Remember, we've got a phosphate group, a deoxyribose, and a nitrogenous base. We see phosphates in mRNA nucleotides as well, all RNA nucleotides. And then adenosine is kind of like adenine, as one of our nitrogenous bases in DNA and RNA, although it has an extra component to it. So it's not, we can't say adenine triphosphate, but it's structurally very similar to adenine. And so what we typically do is draw it in, in this way. We can use something to represent our adenosine, and our triphosphate means that we have three. Oops. Circle too much there. Adenosine triphosphate. So we got 1, 2, 3 phosphates. And that's what our ATP molecule is. Now, what's interesting about ATP is that it can actually, I guess I'll just redraw it here. So we can have our adenosine triphosphate, but if we add water to that, add H2O, we actually can get out adenosine diphosphate plus a lone phosphate plus what was here in this bond right here, this covalent bond, is a little bit of energy. And this is the process by which our body uses ATP to get this energy to function. And if you remember, we use energy in active transport. So think back to movement of sodium and potassium across the membrane of an axon or any other membrane. When your muscles contract, every movement requires the use of ATP, and this is the reaction that happens. So if you think about this, this connects to another section. We have adenosine triphosphate plus water, and it breaks adenosine triphosphate into adenosine diphosphate and a phosphate. So this is another form of a hydrolysis reaction, but the arrow going the other direction would be a condensation reaction, a special kind of condensation reaction, and that's in fact what we're going to be talking about. We're talking about how we make lots and lots of ATP so that we can then break that ATP down into ADP, a lone phosphate, and some residual energy. Now sometimes this energy can come off as heat, and that's good for us because we need to maintain a stable body temperature. Think back to the negative feedback loops of being too hot or too cold. We need to make sure that we release this energy, and sometimes that energy gets quote unquote wasted as heat. But if we think about our enzymes, it is important that we keep our enzymes at their optimal temperature, and the way that we do that is through the cellular respiration and burning off that heat is energy. That energy is heat. Okay, well that being said, going back to the targets. Oh, yeah, I'll leave it there. Okay, so going back to the targets, actually, I'm not quite ready to move on. I want to talk about what cellular respiration is and give you an equation for it before we talk about aerobic and anaerobic cell respiration. So you should know that, I'll use a different color to make it a little more distinct. So cellular respiration, you should know the standard formula, which is C6H12O6 plus O2 gives us CO2 plus water plus lots and lots of ATP. Now, this is the overall equation, but let me just point out that this is glucose, and glucose is usually the molecule that goes through cellular respiration. Remember, glucose gets into our body stream at the small intestines, gets into our blood. There's a homeostatic level of glucose that insulin and glucagon regulate, but that glucose gets around your body into the cells, into every single cell for this reaction. But if there's not enough glucose, let's say you're deprived of food, your body's going to use a different molecule, throw it in the pathway of cellular respiration, and get as much ATP out as possible. For example, fatty acids can be thrown into this equation. Fatty acids are modified in this process, but they go through this same thing as a secondary source of chemical energy from this organic molecule. And even if there aren't fatty acids, then we go to amino acids, and those amino acids get modified and go through this process to make ATP to continue surviving. So, in essence, what someone would be doing is digesting their muscles in order to continue to make energy to survive. Okay, so let's actually talk about aerobic respiration. So number 4 first. An aerobic respiration requires oxygen. Aerobic means oxygen. Think of aerobics exercises. You're really concerned about the amount of oxygen that you're getting. You're controlling your breathing. It's about oxygen. So if we didn't have oxygen, it'd be anaerobic respiration. So let's talk about aerobic respiration first. And I'll point out that aerobic respiration requires so it requires oxygen and the use of a mitochondria in eukaryotic cells. And so that's what we're talking about first, is this idea of the mitochondria. And so we haven't talked in great detail about this, but this is when you really need to learn about it. So the mitochondria I'm going to draw it as an oval, has this lining in it that looks like this. It's got an inner membrane that looks like that. There are a couple key parts here. The inside is called the matrix. This line in here we'll call the inner mitochondrial membrane. And this other one on the outside is the outer mitochondrial membrane, but I'm not going to label it yet. So we're going to actually first focus in here at the inner mitochondrial membrane. But let me just tell you that there are 3 parts to this reaction, and I guess I can just list them up here, and we'll come back to them and talk about them in order. So the third one, which we're getting ready to talk about, we're doing this backwards here, is called the electron transport chain. The second one is called the Krebs cycle, and before that is glycolysis. So for aerobic respiration, all 3 of these have to happen, but really the aerobic part, the part that requires oxygen, is number 2. Well, really it's number 3, but number 3 depends on number 2. Number 2 depends on number 1. But number 1 can happen even without oxygen. So, and again, we'll talk about that. This is, I guess I should just erase that. Okay. So we're talking about the electron transport chain here at the membrane of the inner mitochondrial, of the inside of the mitochondria. And sometimes we refer to this as the cristae. The cristae of the mitochondria. And remember, when I say the mitochondria, I'm talking about one of many inside of every single cell in your body. All right, so let's talk about the electron transport chain. I guess I'll just scoot over here. All right, so I'm going to draw, I'll tell you that this is a simplified model of the electron transport chain, but it is a little bit beyond what IB requires of you, but I think these details are going to help you make some sense of what's going on and be able to apply these ideas. Excuse me, if you really understand them and know them, you should be able to apply them to answer the IB question. So even though IB says you don't need to know these details, you do need to know these details to be successful. So what I'm drawing here is a phospholipid bilayer. So I hope you recognize this from 9th grade bio. This is a cell membrane. So this is also what interestingly, we'll come back to this in the future, what the inner mitochondrial membrane looks like. So this is the inner mitochondrial membrane. And let's say that this is the intermembrane space. So this is space between the 2 membranes, and over here on this side is the matrix of our mitochondria. This ETC is the electron transport chain. I won't continue drawing too many more phospholipids. You can fill in these phospholipid tails if you like. You can just keep adding, and know that there are 2 per phosphate head there. And then what I'm going to draw here is, as best as I can, a protein, and it's usually drawn in this shape, that we will label as ATP synthase. So we know it's an enzyme. It's got ASE and it makes ATP. It synthesizes ATPs. So what ends up happening here is that ATPs are made out of ADPs and a lone P. They get this ATP synthase puts them together to make lots and lots of ATPs. But the way that this protein works is that it needs something to turn it. It needs a gradient on which to turn that. And really what it comes down to is hydrogen ions that flow through the ATP synthase in that direction. And the way that they flow through diffusion is that's only going to happen if we pump the inner membrane space with lots and lots of hydrogens a lot more than what's on the inside or in the matrix. So that's what the electron transport chain does. So I'm going to leave this as a box. There's a lot of other details that go into the electron transport chain that I'm not going to go into because there's a lot of detail and we're going to try to focus on the larger picture here. But in order to get these hydrogens to diffuse through the ATP synthase, we need to create a concentration gradient, and hydrogens get pumped out. So we'll end up with lots and lots of hydrogens out here, and just a few on the inside. So what we would say is that there's a high concentration in the inner mitochondrial membrane, and in the matrix, there's a low concentration. And that's what the electron transport chain essentially does. And in doing so, it is creating this, it's allowing this ATP synthase to function. So I did tell you that this is part of aerobic respiration. So oxygen is at play here. And so the oxygen that we breathe in through our lungs that go through the alveoli because of the concentration gradient diffuse into the bloodstream in the capillary beds that surround the alveoli and travel around the body, diffuse into our cells through the cytoplasm into the mitochondria. And it's here that the oxygen combines with this hydrogen in a different, not in the same ratio, but in the end to produce a byproduct of water. And that H2O is very, very important to us. We use it for the aqueous solution in which our proteins fold. We use it for hydrolysis reactions. We use it to provide some volume to our cells, for diffusion of solvents around the body, to regulate our body temperature, lots and lots of different things. And so it's very important that we make this, but it's essentially a byproduct because our goal of cellular respiration is really this molecule right here, ATP. Okay? So this oxygen and these hydrogens combine with one another here at the electron transport chain. And they're really coming to the electron transport chain in a molecule, in a couple molecules, FADH2, so there are two hydrogens that are coming on this, but really only one of them goes, and NADH. Now, these two molecules were made in the Krebs cycle. Remember we talked about the Krebs cycle? So we've talked about electron transport chain. We make lots and lots of ATP and water here. But now we need to talk about the Krebs cycle, which happens in the matrix. So we will now talk about that. So I'm just going to scroll down here. And what's happening, okay, so let's label this as, we're talking about the Krebs cycle. It's named after Mr. Krebs, who discovered this reaction, or series of reactions. And it's also known as the citric acid cycle. And you should be able to recognize both names. So in the Krebs cycle, a couple of interesting things are happening. So there are a couple of, so remember that this is all happening, let's remember that this is happening in the matrix of the mitochondria. And we already know that the products of this reaction are FADH2 and NADH. And these two molecules, remember, carry these hydrogens to the electron transport chain. The other thing that they're carrying are electrons, and those electrons are used as an energy source for that concentration gradient to make ATP. Now, there are a couple things that come into this reaction, and the first of these is something called pyruvate. And pyruvate is a byproduct of glucose, but that pyruvate in the matrix gets broken down into these molecules and carbon dioxide. And carbon dioxide is therefore considered a byproduct. Even though we seem to focus mostly on that in our reaction, carbon dioxide is a byproduct of this reaction, and so that's what we breathe out. So we talked about just a minute ago breathing in oxygen for electron transport chain for cellular respiration. Carbon dioxide is a product that is made here in the Krebs cycle and also part of the glycolysis reaction, or immediately after glycolysis. And we have to get rid of it. And so this diffuses back into our blood. The hemoglobin, the red blood cells that brought the oxygen to our to each cell, will carry the carbon dioxide back to the alveoli so that we can get rid of it through exhalation. Okay, so that's all that you really know, but it's really pyruvate that we need to then figure out where we got this from. Now, pyruvate is something that we get from I can spell, I promise. Glycolysis. If we break that down, lysis means to break down. Glyco refers to sugar. Think glucose or glycogen. Glyco means sugar. So there's another way of saying sugar. So this is the breakdown of our glucose molecule. And glucose, which, if you remember, is C6H12O6, glucose actually gets broken down into two pyruvates. So this is another pyruvate. And so if we do the math, we know that pyruvate is actually a 3-carbon molecule that then proceeds through the Krebs cycle. And that's the process. So glycolysis doesn't happen in the mitochondria, but instead happens in the cytoplasm. And because every single cell has cytoplasm, all living things can do glycolysis. Only things with mitochondria really, and there's an exception to that rule we'll talk about in the future, can do aerobic cellular respiration. But glycolysis doesn't use any oxygen, so this is not considered aerobic. It's actually not even considered anaerobic because there's no need for oxygen in this reaction. Because it happens in the cytoplasm, all living things can do this. And there is another byproduct of this, and it's a few ATP. It's just really 2 ATP molecules that are gained from this process. And you've got some diagrams in your book, page 79. I'm looking at the bottom of page 79 that shows this reaction, but really only a few ATP molecules are made. So bacteria that don't have mitochondria, that perhaps are not doing Krebs cycle and electron transport chain, are making a few ATP per glucose molecule. We, having mitochondria, are able to get a lot more ATP out because the majority of the ATP molecules are coming out of the electron transport chain. So we're able to go through that process as long as we're able to get oxygen into our body. So if we're not able to get oxygen into our body, we run into a separate issue of anaerobic respiration. Anaerobic respiration. So anaerobic means without oxygen. So our reaction here on the, at least the reactants, are just going to be glucose in the normal setting, and there's no oxygen. And in humans, I guess I can just write this down here. This is for humans, and other animals, in fact. We make something called lactic acid. And you might be familiar with this. You might be familiar with the burn that is associated with this event, especially if you're an athlete and you run for a long time and your breathing is not as controlled. You're not getting as much oxygen to your lungs, into your bloodstream, into your mitochondria. Your body is triggered to stop doing electron transport chain, to stop doing Krebs cycle, and instead of converting that glucose into pyruvates, it converts into lactic acid, which is a 3-carbon sugar, which, interestingly, when your body gets more oxygen, so think about what happens when your body starts to burn. You stop running. You're supposed to stop, catch your breath, and get oxygen back into your body. And when that happens, this lactic acid, which is stored as an acid, which causes the burning, gets reconverted back. Well, I guess I should point out there's an intermediate. So this glucose goes to pyruvate because it still goes through glycolysis, but instead of going to Krebs, it gets converted to lactic acid. So this bottom part is a more complete process. And when you have oxygen, this lactic acid gets converted back to pyruvate. Remember, it's also a 3-carbon sugar. And then the pyruvate goes through Krebs and etc. So lactic acid is kind of like a storage of it's a storage of the carbons until your body can process them. But that's not the case for all organisms. And so the other one that we need to talk about is yeast. So yeast is a fungus. I hope you know that. It's a single-celled fungus. Yeast does a very similar process. It does need sugar. So think about yeast that you use to bake bread. You put yeast into, let's say, flour. Flour, by the way, is mostly starch, which is a polysaccharide. Yeast release enzymes outside of their cell to break down those polysaccharides to monosaccharides. So they hydrolyze those polysaccharides, they absorb those monosaccharides, and they use them for respiration. So yeast can do aerobic and anaerobic respiration. When you put yeast into your flour with some water, they produce bubbles, some foam, and that's actually carbon dioxide that they're producing through respiration. But they can actually produce carbon dioxide from two different ways. So one, they can follow the exact same pathway that we do, the one that we followed with glycolysis, Krebs, and electron transport chain, but in the absence of oxygen, or in yeast's anaerobic respiration, that glucose gets converted to pyruvates, those 3-carbon sugars. That pyruvate gets broken down into carbon dioxide, which is the bubbles, and ethanol. And ethanol is a 2-carbon sugar. So we've got 2 carbons here and 1 carbon here for every pyruvate that was broken down from this glucose molecule. And this carbon dioxide is lost in the environment. So really, it can't reverse. And a 2-carbon ethanol cannot convert back into a 3-carbon pyruvate. And so this reaction can't reverse, unlike the anaerobic respiration that happens in mammals. And in fact, this is the ethanol that is alcohol. So if we think about, if you know sources of alcohol, if we think about barley, we think about wheat, think about grapes, for wine. We think about potatoes for vodka. We think about, any sugar cane for rum. All of those things have a sugar component that then gets combined with yeast to ferment. That's this process right here. What we're talking about in anaerobic respiration yeast is fermentation. It ferments to make carbon dioxide and ethanol, and the reaction cannot reverse once ethanol is made. Okay? And so that's really, I think that's it. So you need to make sure that you understand. Let's see if we can go back to... So you should be able to define cellular respiration, understand ATP, being able to draw them and explain where the bonds are is a fantastic model. If you can make sure you can do that. You need to be able to outline aerobic respiration, so that's Krebs and electron transport chain last and then Krebs, and glycolysis. Glycolysis happens at the start of both aerobic and anaerobic respiration. But anaerobic respiration happens in the absence of oxygen, and in animals or in mammals it does something very different than yeast. And I think that wraps it up. I would recommend that you look at the few questions that are at the back of this section on page 83 at the very top of the page, and make sure that you can answer those questions either from the reading or this podcast. And if you can't, come to class with those questions and we'll talk about them, okay?